today I want to talk to you about a massive shift on the ground in pharmaceutical drug development. And I called my talk Beyond AI, simply because that's probably one of the major driving forces of technology today. We are all using it, right? Claude, ChatGPT, Gemini, we're using it in every single aspect of our lives, and it's very clear to us that it's pushing the industry forward. The question is, how far can it, and how can it make it better? How do we change the industry with those new forces? And let's talk about it. So we have a problem in pharmaceutical drug discovery. The old model, the old world is dying away and the old models are breaking. And it's been going on for a while. It used to cost about $500 million to bring a single drug to the market. But today, we're talking about an average of $2.5, $2.6 billion to bring a single drug to the market and more than a decade of work. This is driven by massive level of attrition in clinical studies. More than 90% of clinical studies and subsequent market approval in phase 4 fail. And the industry is spending $250 billion every year to push this forward. And those $250 billion are only generating less than 50 drugs every year that are approved by the FDA. Some of them are old drugs, right? Our level of innovation has been decreasing for a while. It's the inverse of Moore's law. And one of the reasons this is happening is because we know we've been using the wrong models. Mice are cheap, mice are easy, and they've been the go-to model for the past 100 years or more. But it's important to understand just how bad of a model a mouse is. You know, it's not a small human. We know that mice have smaller brains, but they have a completely different structure. They don't have folds, right? Our heart rate is different from that of a mouse by an order of magnitude. A mouse heart is closer to our brain than our actual heart. It runs on sodium and glucose. Our breathing rates are different. 7-fold difference between breathing rates between human and mice. 2-fold difference in glucose concentration, differences between triglycerides and cholesterol as primary lipids. We're talking about completely different approach to inflammation. We are using the wrong model, and it shows. Because about 70% of the drugs that are toxic to humans are not toxic to mice and vice versa. Just by a show of hands, how many people have been taking aspirin in this room? Great. It's a great rat poison. It works well on every one of us. And this is why there's been a growing revolution that has been driven by literally everybody in the field. Not only Republicans, but also Democrats. The FDA Modernization Act has taken the United States by storm, and very recently the European Commission issued their own roadmap. They're talking about new approach methodologies, NAMs. The idea is simple. We know that mice don't work all the time. They are not extremely reliable, but they're the best we have. So the change that happened essentially now is saying it's when you add human on a chip, AI predictions, organoids, they're not end. They're not a yes end to an animal studies. You can make the argument today that they are instead. And what they're looking to see are context of use, explain why your model fits the problem you're trying to solve. They're looking to see that it's human relevant. It's coming from human cells or clinical data. And last but not least, that it's reproducible. One of the biggest hurdles right now in the field. But if you get that, you can actually go to an IND in clinical studies without passing through animals. And we're talking about guidelines that have been shared by the FDA just in the last couple of months, and a roadmap that have been issued by the European Commission just in the last few weeks. But these are the tailwinds, and they're changing fast. And this is the point where everybody has been betting big on AI, has done exactly the same bet when it comes to pharmaceutical drug discoveries. So it's been $1 billion of investment in the idea that we can take those large language models or these complex trees and train them on the genetic data that we have available and chemical space that we have available to predict new drugs. And some of it has been extremely successful, right? Isomorphic labs in silico medicine have done amazing work on actually reshaping target discovery, molecular design, and doing it in days rather than months. So we have new drugs coming. But there is a problem. The clinical translation is just not there yet. In the recent studies, 2023, 2025, in phase 2 studies, they are doing no better than everything else. And the problem is basic. We don't have the right data. The data that we have is completely non-reproducible. 85% of papers today, everything you train your AI on is false. You can't repeat it. And that means, imagine that you're trying to build Waze, you're trying to guide a car somewhere, but the maps are wrong, right? So where are you going? And this is where other methodology came from a very different space. Right? Hans Clevers and others have been developing human organoids for the last 2 decades. These are human cells that essentially grow into a tissue. And whether it's a beating heart, a human kidney, or even a human brain, we can actually simulate that really, really well. And they capture human physiology, human genetics, and human metabolism in a way that mice can't. The problem is that these are beautiful artisanal creations. They vary from day to day. They vary between technicians. They vary between labs. And the variability sometimes can get to 70 to 90%. So reproducibility is simply not there. And this is where we think the field is heading. Because one of the things that my company, Tissue Dynamics, we've been doing at the Hebrew University, and other groups have done around the world, is try to use robotics in this new biology. And we developed Rodeo. So this is robot-directed organoid deposition, the ability to use robotics to essentially deposit single organoids in single wells on exactly the same size, with exactly the same metabolic function, with exactly the same cellular composition. And when you do that, you move from 80% variability to less than 20% variability. So now you can actually ask questions and expect to get the same answer in the same time. Otherwise, organoids are kind of Jewish. You ask 3 organoids 1 question and you get 4 answers. Nobody's laughing. Okay. And these robotic systems can run tens of thousands of human organoids at the same time. And what it means is that your data starts looking like this. Every line is a different human organoid responding to a different drug. This is very, very robust, and it gives you dense, reliable human physiological data that is at the machine scale, right? We're talking about tissue dynamics running with IA funding over the last couple of years, more than 30,000 human organoids, 40 million data points, and over 200 compounds, creating a massive database of human physiology that nobody else has, right? And companies like Vivodyne are running to do exactly the same thing, and Roche is investing billions of dollars in building similar technologies right now in Switzerland. Now, once you have data like this, you can look at human physiology in a way that nobody else can. Because it's no longer endpoint data that you're looking at TC50 curves and asking, okay, I'm giving a drug, I'm waiting a week, and then what happens? Is it alive? Is it dead? No, 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 no. We're seeing physiology. We're seeing biomarkers changes. We can actually extract features that are spectral. We can see variability as a function of time. We can see dynamics, and that kinetic data allows you to understand the mechanism of action in the way that nobody else can. So when you give us your drug, we're not telling you it worked or it didn't work, but how it worked. and better. How to make it work better? And this leads to a vision that has been shared by major corporations and companies, the idea that now, this is something we can train AI on. This is the concept, the vision of lab in the loop, the idea that you can design and build a system that generates data, test your drug, test your disease in organoids, then use that data to learn, have the agents make a decision, and repeat the experiments. And now, instead of having 10, 20 scientists in the lab, right, you have 100,000 AI agents running experiments in parallel in a room that is smaller than the shower in your house, right? So things can actually accelerate, and they accelerate extremely fast. So let me show you what we can do with this. So one easy problem, relatively easy, is human safety predictions. This is where the ground truth is known. The FDA actually had cardiotoxicity and liver toxicity data published. And AI models are doing okay, about 75% productivity of safety of drugs, toxicity in the kidney and toxicity in the cardiac, in the heart and in the liver. When we use our data, when we actually drive it with human organoid data, the numbers are truly different. Using kinetic data, we're getting closer to 80% productivity, and using AI linked to that data, we're actually getting to 96% productivity in liver injury and 88% productivity in drug-induced cardiac toxicity. So numbers that are about 15% better than all the models published to date. And we can take that, and not only take the data, but actually show that it translates to the clinic, make decisions in the clinic. Most recently, we published a major paper that showed that our AI models can actually predict nephrotoxicity in oncology medication. We can understand the medication, we can understand the mechanism, and then reverse it, block it, and allow you to improve your clinical study. In a retrospective clinical study of 247 patients, we can actually show that we can drive decision-making better than any other model. We can also take the same technology and model disease progression. This is the world's first model of human sporadic Alzheimer, not genetically induced, but actually taking human organoids, running them for 200 days, almost a year, in our robotic system, and showing you can get phospho-tau, amyloid deposition, secreted biomarkers, the same pathology of human Alzheimer in a dish. And not once, hundreds and thousands of times, allowing you to look at drugs. And this is not, I have 1 minute, and this is not just disease development in a single organoid models. Your models can be complex. A new model for COPD, we're talking about vascularized lung organoids that can actually integrate neutrophils into the assay itself. We can look at dynamic, complex immune response in a dish, in a robot, and do it at scale. And you can take this and dream of only what you can achieve. This is a human organoid that has been developing for more than 100 days in the lab. And you can see for the first time, not only GABAergic signaling, but also alpha and beta waves. This human organoid stopped dreaming and started waking up. Now we have a $2.2 million from the Templeton Foundation, actually looking at fundamental mechanisms of human consciousness on a dish in a way that we never thought was possible just a few years ago. My last slide. Imagine what you can do with such a system, because we are all thinking the same thing. Can I use it to predict the future? And this is a complex question, but imagine what you can do in NASDAQ if you have 80% accuracy in predicting clinical failures. This is, by the way, what Prof is doing, a new spin-off company of tissue dynamics, and it's very, very exciting to see how the entire field is moving. So, last slide. This is a new market. This is a new decade. This is a new century. This is where the field is going. It's not only artificial intelligence, it's a combination, the intersection of human organoids, robotic automations, and technology that is, some call bioconvergence, some call bioengineering, but the winners are not going to be people that are doing software alone. The winners are going to be the people that can generate new data.